A 200-year-old hair sample matched a person alive today 2 centuries ago. A single strand of human hair was sealed away, untouched, unaltered, forgotten by time. Today, that same strand has spoken again. Not with words, but with something far more precise. A genetic signature. When scientists extracted its DNA, they expected fragments, damage, maybe even nothing at all. Instead, they found something complete enough to compare. And when they ran that comparison against modern databases, the result did not point backward into history, it pointed forward. To someone alive right now. Not a distant relative. Not a shared lineage stretched across generations. But a match so close, it blurred the line between identity and coincidence. According to everything we know about human genetics, this should not happen. DNA is not static. It shifts, it evolves, it carries the marks of time like fingerprints across generations. And yet, somehow, a biological signature from 200 years ago appears to exist again, in the present. The question is not just how this happened. The question is, what exactly was found inside that strand of hair that refuses to stay in the past? In the early 1800s, preserving hair was not unusual. People kept locks of hair as keepsakes, memorial tokens, or even scientific curiosities. This particular strand, however, was different. It was catalogued, labeled, and stored under conditions that allowed it to survive far longer than most biological material ever could. Over time, it became just another artifact, an object tied to a forgotten individual whose name carried little relevance in the modern world. For nearly 2 centuries, it remained silent, offering no reason to be revisited. Until recently. With the advancement of genetic sequencing, scientists began re-examining historical biological samples. Hair, once thought to be unreliable due to the absence of a root, could now yield mitochondrial DNA and, in rare cases, even fragments of nuclear DNA. This opened a new door into the past. Researchers could reconstruct lineages, confirm identities, and connect historical records with biological evidence. It was a method grounded in science, built on probabilities, and constrained by known limitations. Nothing about it suggested the possibility of a direct match with a living individual. When the analysis of this 200-year-old hair sample began, everything followed expected patterns. The DNA was partially degraded. Certain sequences were missing. Others were intact enough to form a usable profile. The team processed the data carefully, filtering out contamination, verifying authenticity, and reconstructing as much of the genetic code as possible. It was meticulous work, but routine by modern standards. At first, there was no indication that this sample would be any different from countless others analyzed before it. Then came the comparison. Modern genetic databases contain millions of profiles. Most matches are distant third cousins, fourth cousins, shared ancestry from centuries ago. Even close matches, like siblings or parents, follow predictable patterns. But when this ancient profile was entered into the system, the result did not behave predictably. It didn't return a list of distant relatives. It returned one result. A single individual. Alive today. And the match was not partial. It was not distant. It was, by every measurable standard available to the system, nearly identical. At first, the result was dismissed as an error. Contamination is always a possibility in genetic analysis. Modern DNA can easily overshadow ancient samples. The team reran the tests. They isolated the sample again, repeated the extraction, and processed it using independent equipment. The outcome remained unchanged. The same modern individual appeared. The same level of similarity. The same statistical improbability. This is where the mystery begins to deepen. The simplest explanation was that the living individual was a direct descendant of the person who owned the hair. But genetics does not work that way. Even between parent and child, DNA is not identical. It is recombined, reshuffled, altered slightly with each generation. Over 200 years, those changes accumulate. Mutations occur. Variations spread. The idea that two individuals separated by multiple generations could share an almost identical genetic profile challenges the very foundation of how inheritance functions. Another explanation suggested that the database itself contained errors. Perhaps the matching algorithm was flawed. Perhaps the reference data was incomplete. But when the profile was tested across multiple systems, using different methodologies and independent datasets, the result persisted. Different labs. Different researchers. Same outcome. One living person, matching a 200-year-old biological sample with a level of precision that should not exist. This led to a deeper question: what exactly does a match mean? In genetics, a match is rarely absolute. It is a measure of probability, a comparison of sequences, a calculation of similarity. Identical twins are considered genetic matches because they originate from the same fertilized egg. Outside of that, perfect matches are not expected. Even clones, in theory, would accumulate differences over time due to environmental factors and random mutations. So when scientists say that this modern individual matches the ancient hair sample, they are describing something that approaches a level of similarity typically reserved for identical twins. But identical twins cannot exist 200 years apart. One theory proposed that the match was not as exact as it appeared. Perhaps the degraded nature of the ancient DNA created gaps that were filled in by the algorithm, leading to an overestimation of similarity. This is a known issue in genetic reconstruction. Missing data can sometimes be inferred, and those inferences can introduce bias. However, when researchers focused only on the confirmed, high-confidence segments of DNA, the similarity remained unusually high. Not perfect, but far beyond what would be expected across centuries. Another theory explored the possibility of rare genetic stability. Certain regions of DNA change very slowly over time. These conserved sequences can remain nearly identical across many generations. If the analysis focused heavily on these regions, it might create the illusion of a closer match than truly exists. Yet, even when variable regions were examined, areas of DNA that are known to change more rapidly, the pattern held. The modern individual still aligned with the ancient sample in ways that defied standard expectations. A more unconventional theory began to emerge, one that moves beyond traditional explanations. It suggested that under very specific conditions, genetic information could be preserved and reintroduced in a way that mimics continuity. Not through direct inheritance, but through mechanisms we do not yet fully understand. This is not a widely accepted idea. It sits at the edge of scientific speculation. But the data, however incomplete, leaves space for questions that current models struggle to answer. Then came a breakthrough moment. A collaborative study involving multiple institutions took a closer look at the structure of the DNA itself. Not just the sequence, but the patterns within it, markers, repetitions, and subtle signatures that can reveal deeper information about origin and change. What they found added another layer to the mystery. The ancient sample contained specific markers that were considered rare even in modern populations. These markers appeared in the living individual as well, in the same configurations, with minimal variation. Statistically, the likelihood of this occurring by chance is extremely low. This discovery shifted the conversation. It was no longer just about similarity. It was about specificity. The kind of specificity that suggests a direct connection, even if the nature of that connection remains unclear. Researchers began to consider scenarios that had previously been dismissed. Could there be undocumented lineages that preserved genetic material with unusual fidelity? Could certain environmental or biological factors slow the rate of genetic change? Or was there something else entirely at play, something that current science has not yet defined? The twist, however, did not simplify the mystery. It made it more complex. Further analysis revealed that while the overall genetic profile was strikingly similar, there were subtle differences. Small variations that indicated the two individuals were not, in fact, identical. These differences were consistent with the passage of time, but not at the scale expected over 200 years. It was as if the genetic distance between them had been compressed, reduced to something far smaller than normal inheritance patterns would allow. This raises a deeper question about how we understand identity at a biological level. We often think of DNA as a fixed blueprint, a code that defines who we are. But in reality, it is a dynamic system influenced by countless factors over time. The idea that two individuals could share such a close genetic identity across centuries challenges not just the science of genetics, but the concept of individuality itself. It suggests that under certain conditions, biological information can echo through time in ways we do not yet fully comprehend. Imagine discovering that a part of you, something as fundamental as your genetic code, has existed before. Not in a vague, ancestral sense, but in a form so similar that it appears almost duplicated. It changes how you think about history, about inheritance, about the continuity of life. It blurs the boundary between past and present, between origin and outcome. And yet, the most compelling aspect of this story is not the science. It is the unanswered question that remains at its center. Why does this match exist? Despite advanced technology, repeated testing, and extensive analysis, there is no single explanation that fully accounts for the data. Each theory addresses part of the mystery, but leaves gaps. Each discovery adds detail, but also introduces new uncertainties. The more we learn, the more the original contradiction remains intact, a 200-year-old strand of hair, and a living person today, connected by a genetic signature that should not persist across time. In the end, this is not just a story about a scientific anomaly. It is a reminder of how much we still do not understand. Biology, for all its precision, still holds complexities that resist easy answers. The past is not as distant as we assume. And sometimes, it leaves traces that reach further into the present than we are prepared to explain. Because if a single strand of hair can carry a genetic identity across 2 centuries, then the question is no longer just about how this happened. The question is what it means for everything we think we know about where we come from. And how far that connection truly extends. How far that connection truly extends. Researchers found the DNA differences were too small for 200 years, almost as if the same genetic pattern had quietly reappeared instead of evolving normally. And when similar fragments showed up in a few unrelated people, the mystery shifted. Maybe this wasn't inheritance, maybe it was recurrence. If the past can echo itself through DNA, then identity may not be as fixed as we think. Like the video, subscribe to the channel, and share your thoughts, because some mysteries only get deeper the more we look.